Hello, everyone. Welcome to our PacVec Fall Seminar Series. Today, we have two great talks from a training grant recipient and a graduate student. If you have any questions during the talks, please hold them or type them into the chat because Dr. Tara Tiemann from University of Pacific will be facilitating the Q&A at the end of each talk. First up, we have Kirsten Meredith. She is a PhD student in the School of Biological Sciences at the University of Utah with an interest in host discrimination and other odor-mediated behaviors in mosquitoes, as well as their flight dynamics. Kirsten has a Bachelor of Science in Biology from the University of Utah, and since insect vector biology intersects with many aspects of society, Kirsten's main goal is to take what she learns from her PhD and apply it to the interface between science and politics, education, business, as well as outreach to the general public. So with that, Kirsten, you may share your screen.
All right, can everybody hear me all okay? Fantastic. Let's see. This one. All right, can everybody see the presentation all right? Awesome. Okay. So as Celia mentioned, I'm Kirsten Meredith, and I'm a fourth-year grad student at the University of Utah and prior recipient of a PacVec training grant in 2020. I want to share with you some of the research funded by the grant and further endeavors in our lab. Here's a quick overview of my talk. I will introduce my research aims and review specifics about the Western treehole mosquito. I will mention the paper that inspired part of our research and go over our experiment and findings. I'll then transition to our other primary investigation and the supporting fieldwork. And then lastly, I'll show you how we intend to use said fieldwork to investigate novel flight mechanisms in mosquitoes. For my research, I am interested in the nuances of olfactory-mediated behaviors within and across species of mosquitoes. Chemosensory-informed behaviors can have a variety of nuances. For example, host-seeking odorant-mediated flight involves mosquitoes reacting to ephemeral odor cues to locate a source. This small video on my slide shows the end of such a flight in a wind tunnel. Other behaviors, such as site selection for oviposition, can be enhanced by odorant molecules, but the sensation of these molecules does not specifically induce egg-laying behavior. Thankfully, many groups are studying the most concerning human-centric species, and their work presents an opportunity for me to make valuable comparisons between those species of most concern and others that are related but remain less studied. My current work involves the local nuisance species Aedes sierrensis, or the Western treehole mosquito. This species bears many similarities to Aedes aegypti and Aedes albopictus. It's a container breeding species that vigorously bites mammals and humans and lives adjacent to human habitats. While it does not seem to be a primary vector for any human arboviruses, it does transmit the dog heartworm. Due to its opportunistic and aggressive feeding behaviors, mosquito control communities have significant interest in understanding Aedes sierrensis. Studying Aedes sierrensis can inform us about Aedes aegypti by contrasting their differences. Aedes sierrensis is adapted to more temperate climates with winter freezes, while Aedes aegypti is adapted to warmer, drier climates and is not tolerant of winter freezes. This contrast is especially poignant here in Utah. The Wasatch Front region, vaguely marked out by that red star, is one of the most southeastern points of the Aedes sierrensis natural range in the state. Southern Utah is typically too dry and lacks the canopy tree cover that Aedes sierrensis prefers. However, mosquito abatement districts in southern Utah monitor intensively for Aedes aegypti, and opportunistic invasions of aegypti have been found and suppressed in St. George and Moab. Observations suggest that all incursions of aegypti into southern Utah have been human-mediated, i.e., they're hitchhiking in, likely on vehicles traveling in from established Aedes aegypti locales further south. Ultimately, these two species represent a local niche dichotomy for Utah. Where one exists, the other usually doesn't. I'll come back to this point later on. So in interest of contrasting these species, our lab found an interesting paper. Recent research by Mello et al. In 2020 presented a novel avenue for controlling and mitigating Aedes aegypti mosquitoes. They were able to show that gravid female mosquitoes are attracted to geosmin, an odor reminiscent of wet earth. Furthermore, they demonstrated that beetroot peel is a cheap and easy way to substitute for pure geosmin in gravid traps. Given their success, we wanted to see if these same patterns held for Aedes sierrensis. Gravid sierrensis mosquitoes will readily lay eggs in dark containers lined with egg sheet paper and filled with plain distilled water. We wanted to see if our Aedes sierrensis mosquitoes showed a similar preference for beetroot peel infused water compared to plain water. From another paper by Matthews et al. In 2019, we know that Aedes aegypti mosquitoes sample the water near the intended egg-laying areas to screen for factors such as salinity. To contrast the beetroot infused assay, we ran a second assay that gave gravid mosquitoes a choice between water and larva rearing media. The larva rearing media is a nutrient broth with brewer's yeast in water, a common choice used in mosquito rearing protocols to provide Aedes sierrensis with the appropriate deoxygenated but nutrient-laden environment to stimulate egg hatching and early larval growth. We hypothesized that female mosquitoes would prefer both the beetroot infusion and the nutrient broth mixture against plain water, as both offered better nutrient profiles for their offspring. We set up a two-choice oviposition assay with water as the control and either broth or beetroot peel infusion as the alternative. We ran 7 assays with the broth and water and 10 with the beetroot and water. All our experiments were initially run in parallel, with the locations of the egg-laying containers varied within the cages. We collected egg sheets after 48 hours. Initially, we counted these eggs under a microscope. They're very, very small. Eventually, we switched to counting them using image J to mark eggs individually from a picture. This modification required washing all the eggs onto a white filter paper, then photographing them on a uniform background for best resolution. This process also helped remove residue and gunk from the beetroot preparation that could be mistaken for an egg. We analyzed these raw counts using the oviposition index from the Mello paper. A positive index indicates the treatment accumulated more eggs than the control, and a negative index indicates the opposite. Our results were quite interesting. For the beetroot assay, we've confirmed that Aedes sierrensis significantly prefers beetroot infused water. The oviposition index is 0.59, and the p-value in a two-way t-test is significant. Like Aedes aegypti, the geosmin produced by the beetroot peel seems to attract the females to lay their eggs preferentially. So just to indicate, these two graphs are all the eggs across all the trials versus the eggs per female per trial. Next. The larval rearing broth actually had nearly the opposite effect. The gravid female mosquitoes seemed to prefer plain water consistently. The oviposition index was a negative 0.52, meaning that they preferred the control almost all the time. But high variance kept our analysis from being significant unless we had predicted mosquitoes would choose the water over the broth, which we did not. We did not run any comparisons between beetroot peel infused water and larval rearing broth, but we would expect these preferences to remain in favor of the beetroot. While gravid mosquitoes would leave eggs on the broth egg sheet, they would consistently leave more eggs on the plain water sheet. Combining what we understand from Matthews et al. And Mello et al., we can insinuate that geosmin seems to increase attraction, and something about the nutrient broth and yeast mixture, potentially a headspace odorant, makes the nutrient broth less attractive to gravid female mosquitoes. We do intend to follow up on these findings, but for now, I want to diverge into the fieldwork portion of our investigation. Odorants like geosmin seem to have a more global effect on mosquito behavior, encouraging certain outcomes, but they are not strictly linked to specific reflexive behaviors like host odor seeking. Geosmin encourages female mosquitoes toward an optimal egg-laying site, but it does not dictate the particular reflex of egg deposition. However, CO2 carried in the air can elicit specific flight behaviors in female mosquitoes. But odorants have to travel in an airflow within any given environment, and mosquitoes must also navigate that environment to use the information carried on the air. These ideas embody the second portion of my research efforts. For Aedes syrensis, there is anecdotal evidence that it is a relatively poor flyer amongst mosquitoes. But what does that mean from an analytical standpoint? Wind speed, turbulence, temperature, humidity can all contribute to the prevailing conditions. There are upper limits for mosquito flight activity if the observations are accurate. There could theoretically be lower limits as well. Flight can be an energetically expensive activity, so it makes sense that mosquitoes could conserve their activity to wait for the right stimuli. So what makes for these optimal mosquito flight conditions, and how do these vary from species to species? To this end, we've been working on a project to characterize the contributing factors that affect mosquito flight activity in the natural environment. So we've been taking some measurements. Using a CSAT 3D sonic anemometer, we've been recording the conditions while simultaneously trapping mosquitoes in a BG Sentinel trap with CO2 lure. The trap included a BG counter, which allowed us to count and bin the relative activity into 15-minute time chunks. We've run these observations in two different habitats, the wetlands west of Salt Lake City and an urban backyard near a creek with known mosquito abatement. We've been able to record several days over the last few years, and each habitat tells a unique story. The wetlands habitat is far more open, and wind patterns can vary quickly with no natural wind breaks. This graph represents one day of recording on July 2nd of 2018. from noon until 8 a.m. the next day. The anemometer can record wind magnitudes in the X, Y, and Z planes at a frequency of 10 Hertz, shown by the bottom graph. The gray lines on these graphs represent raw data. The blue lines represent the 5-minute averages of that data. The area in lavender represents one standard deviation around the means. Turbulence is derived from the wind magnitudes and characterizes how the direction of the wind is changing from moment to moment, represented here by the top graph. The center graph represents the relative mosquito activity as logged by the BG counter, grouped into the 15-minute time bins I mentioned before. Unfortunately, our attempts at getting a more refined time resolution from the BG counter have failed, but the patterns seem to hold. We see certain limits for wind magnitude and turbulence when wild flying mosquitoes are finding our traps. These limits are particularly distinct for the data highlighted here in yellow. This highlight shows the pattern for decreased wind and turbulence with a correlated increase in mosquitoes trapped in an otherwise unremarkable portion in the middle of the night. These histograms here show the distribution of mosquito activity across 50 chunks of both turbulence and wind magnitude and represent all of the trapping days we collected. These graphs help us see the natural ranges of mosquito flight capacity in an open wetland habitat. As indicated by the red line, we catch very few mosquitoes above the 3 meters per second mark, and the drop-off is quite apparent. We're excited because most mosquito research performed in wind tunnels limits the wind magnitude below 1 meters per second. Many flight assays use a wind speed of approximately 0.3 meters per second, as indicated by this green line. This data shows that we have a wide, viable range for testing mosquito flight capacity. Our lab then focused on the urban backyard trapping efforts to find and catch Aedes sierrensis while characterizing aspects of their known local habitat. With information provided by the Salt Lake City Mosquito Abatement District, we were aware that they treated the canopied creek bed just behind our trapping area for Aedes sierrensis tree holes. Unfortunately, we captured no Aedes sierrensis throughout several trapping sessions. This lack of sierrensis is likely due to several significant factors, including local droughts and late season trapping. The Salt Lake City Mosquito Abatement District reported that sierrensis numbers were down across all urban monitoring stations and that it was likely due to the arid conditions of summer 2020. We did capture other species of interest during our trapping and were able to gain a snapshot of the urban mosquito climate. Just like the wetland data, we have turbulence and wind magnitude aligned to our mosquitoes trapped in 15-minute time bins. This trapping occurred during a limited time frame from approximately 8:30 p.m. to 11 p.m. with sunset marked out as shown. But this urban assay shows consistently lower wind magnitudes around 0.2 meters per second throughout the entire trapping session. Turbulence also stayed predictably low. We intend to collect more data during nights where the weather is more variable sometime in the near future. Still, these data show that the urban setting can provide a more moderate habitat for the purportedly poor flying Aedes sierrensis. In summary, open mosquito habitats have a much wider range of flight-relevant environmental variables, even on calm days. In comparison, sheltered urban canopied habitats have a milder range of variables important for flight. Now that we understand some of those variables that contribute to the mosquito flight economy, we can work to unravel them in our wind tunnel. So while some mechanisms of insect flight are well understood, others remain pretty much undescribed. Insect flight experiments have extremely low wind speed and nearly no turbulence, two conditions rarely observed outside. Odorants traveling in airflow in the natural world are subject to a wide range of fluctuations that are usually difficult to replicate in the lab. Our wind tunnel works to address these deficits. Our tunnel is 5 meters in length, humidity controlled, and is quite a bit larger than your standard mosquito flight apparatus. In this tunnel, we have another system installed, which we call the active grid. In collaboration with engineers at Princeton, this series of wireless paddles can manipulate controlled instances of replicable turbulence in otherwise laminar or smooth airflow. This is important because we can begin decoupling wind magnitude from turbulence. This tool gives us new and exciting ways to expose mosquitoes to odorant stimuli while challenging their flight capacity and potentially revealing novel flight behaviors. We're still working closely with our collaborators in Princeton who are fine-tuning the active grid with our field observations. Thanks to an NSF Eager grant, this project is now in high gear, and we hope to be testing mosquitoes by next spring. I mentioned coming back to my earlier point of a niche dichotomy because I wanted to emphasize the importance of having a species like Aedes sierrensis. It represents a certain kind of habitat in Utah that we prefer. Winters that freeze with snow and accumulation of water in trees. With climate change, we're at risk of reducing these habitats and becoming prime territory for Aedes aegypti. The last few winters and summers in Utah have been within a drought. It's important for us to acknowledge that climate change in Utah means we can lose one obnoxious species, but gain a much worse replacement. To that end, I want to thank my lab and committee, our collaborators at Princeton, the Salt Lake City Mosquito Abatement District, and PacVec for the support that these endeavors have required. I'll take any questions now.
Thank you, Kirsten. Very nice talk. If you have questions, you can feel free to unmute and ask or type into the chat. We can start with one that's already in the chat. How was the turbulence calculated or measured?
Right. So the turbulence is derived directly from the given wind magnitudes that we measured. I can share the screen again. Let's see. Right here. It will let me, okay, here we go. I don't have the calculation per se, but the turbulence is being represented here by a factor called turbulent kinetic energy, which is a calculation from each of the wind magnitudes we measured in the X, Y, and Z planes. I did not personally perform this calculation, so I don't know. I am a mosquito person. I have no idea what turbulence is about. We have our engineers to thank for that specific calculation. So if you want more details, I'm sure there's a very nice wiki page on turbulence you can look at, because I have no idea.
Other questions for Kirsten? Another one from the chat here.
Yeah.
Could you remind us, for the oviposition study, did the larval broth previously have larvae in it, or was it a mix created with no larvae?
Yes, it had no larvae. It was freshly made broth that sat 24 hours to let the yeast activate and begin deoxygenating the mixture. It did not have larvae in it. I just chose a very larva-laden picture to get my point across. Let's see, we have one from Benjamin. If you could unmute.
Hello. Also regarding the oviposition study, do you know the water oxygen level in the beet preparation?
No. We didn't measure the oxygen levels of that one because it's important for the egg hatching for it to be deoxygenated, not necessarily for the egg laying.
Okay, cool. Thank you.
Any other questions for Kirsten?
Let's see, I think there's one in the chat here.
Yep, there it is. Can you clarify what it was meant by odorants like, sorry, I don't know how to say it. How do you pronounce it?
Geosmin, yeah.
Geosmin. Encourage certain behaviors, but aren't limited to specific reflexive behaviors.
Right, so geosmin encourages, so you can kind of think about this like, you know, if your mother were cooking in the kitchen, you'd be encouraged to go to the kitchen if you were very hungry, right? You might naturally migrate yourself toward the kitchen, but it's not necessarily going to make you automatically begin eating whatever, you know, you smell. So for flight behaviors, things like CO2 seem to elicit very specific neurophysiological feedbacks that get mosquitoes flying in very specific patterns. One of the most well-known is described as optomotor anemataxis. They turn into the wind with reference to their physical environment with visual feedback. So it's not, geosmin isn't encouraging a specific like reflexively, you know, laid out neurological pathway, but it definitely is encouraging them to get to environments that potentially work for their oviposition behaviors. Yep.
I think we probably have time for one more question for Kirsten. All right, thank you very much, Kirsten. Nice job.
Thank you.
All right, thank you, Kirsten, for your amazing presentation. We'll now move on to our next speaker, Nathan Tsai. Nathan Tsai is an environmental sciences graduate student at the University of California, Riverside, with an interest in contaminant fate and transport, contaminant degradation, pathways, bio and phytoremediation, and environmental waste monitoring and management. His current work sought to characterize the occurrence and distribution of pyrethroids across catch basins in California and in numerous USDS structures in Los Angeles County. Nathan, feel free to share your screen.
Hello. Do I sound okay? Okay,
Yes.
thank you. So let me just share my screen. Let me send it into view. Okay. There we go. Again, thank you, Kirsten, for the excellent presentation. You know, even though I have tried to catch up on my mosquito knowledge, I still don't know if I could call myself a mosquito person, but it was very easy to follow for me. Anyways, again, I'm Nathan from UCR under the JGAN Environmental Chemistry Research Group, right? And today I'll talk about a pyrethroid contamination in California underground storm drain systems. A lot of you vector control professionals in this call are familiar with these storm drain systems, and I'll be talking about their significance and why we are working with them. Before that, I want to give some brief acknowledgments. Again, my lab has given me a lot of experience and a lot of resources to pursue this work. Again, Dr. Jagan, who I'm under, previous postdocs, graduate students, and undergrad assistants who have helped me along the way. Of course, PacVec for all the support through the years, and all of the MVCAC member agencies and their corresponding staff who I talked with. First, I'll talk about some quick background. Again, as probably most of you know, pyrethroid resistance has been detected fairly frequently, at least compared to previous years, right? Recent CDPH study, I'm sure a lot of you have heard Vicky Kramer talking about this, has found increased KDR prevalence in sampled adult mosquitoes. Therefore, we want to, combine our environmental chemistry acumen with, the vector biology knowledge to better understand the dynamics of how this is coming about. And on our end, we are thinking about sources of unintended exposure to pyrethroids and thus selection pressure. Some background on urban pyrethroid transport. For those of you who don't know, pyrethroids are highly hydrophobic substances, even though they are, usually applied in very low volume sprays by the pest control applicators. They can still, once they settle out from those aerosol droplets, they have a strong tendency to partition into things like soil and dust and other solids that have organic matter. Previous work has been done on pyrethroids in the urban environment. A fair amount of work from our lab has studied their occurrence in urban runoff, right? They can come off of concrete with storm water or irrigation water, and they can be retained on concrete for some period of time. So they don't disappear immediately, unfortunately. And also some work by people like Donald Weston has found significant residues of pyrethroids at the other end of the storm drain system, at the outlets and in water bodies. And pyrethroids have been found to contaminate a lot of urban streams across areas like California. So we have previous research at the street level with the urban runoff and at the outlet level with the outlets and the waters of downstream bodies of water. But there is not as much work on the, sorry, there's some weird stuff with the, okay, there we go. There's not as much work on the in between these underground sites, such as these pipes and the catch basins. And these are believed, these are believed to act as a sink for pyrethroids. Vector biologists already know that they're a common breeding source if they are not treated for mosquitoes. And briefly going over some of our previous work, we worked with people in West Valley and Saciolo MVCDs to briefly collect some samples of water and sediment. And we found concentrations in those media comparable to what was found at false and downstream sites. So I'll briefly go over the methods. Again, in 2019, we only worked with 2 districts, so we wanted to increase our sampling range. And thus, in 2020, we collected samples with the help, significant help, from staff in 7 districts: Sacramento, Yolo, Alameda County, Madera County, Delta, Greater Los Angeles County, Orange County, and West Valley again. To collect those samples, we sampled monthly from July through September 2020. Sorry, that's a typo, not 2021, 2020. And a number of basins were selected per region. These were basins that were known by the staff to have a significant amounts of standing water so that we could sample them, right? We did have some issues with basins drying up over the months, unfortunately, but that just happens, especially in a state as dry as California in the dry season of summer. And anyways, half a liter of water was collected with a metal dipper, and in the lab, we recorded environmental data off of that and filtered it into suspended solids. And then we extracted the suspended solids and the water separately to understand the occurrence and the distribution of pyrethroids in those samples. Overall, we got 79 basins sampled in total, again, due to drying over months, sometimes say in August or in September. Some of these basins ran dry, but overall we were able to get data from all 3 months for a good number of those basins, about 58 of those basins. And we got a total from 3 months of 205 water samples that we were able to collect and fully process. We lost a few due to shipping issues, but not too many. I'll go over the results now. Before that, some brief abbreviations just so the figures don't like confuse you with random characters. Again, these abbreviations for the regions just correspond to the region that these samples were collected from, and the pyrethroids have various abbreviations, as can be seen here. We analyzed for 8 different pyrethroids. We also analyzed for another one, tefluthrin, but we removed it from the analysis since it was only used in agricultural settings. In terms of detections, we'll go over concentrations later. 91% of basins contain detectable pyrethroid residues. That is about 180 or so of these 205 samples. These detections decreased slightly over time, but not by a huge amount from month to month, right? In July, we had about 97% detection rate. In August, we had about 94% detection rate. And then in September, we had about a 82% detection rate. And in terms of individual pyrethroids, which I'll go over in a bit more, bifenthrin was by far the most detected pyrethroid. It was found in almost all of the pyrethroid containing samples, 89% of all samples, right? And then other pyrethroids were detected at far lesser rates. These ranged from 0.5% to 24%. In terms of pyrethroids that you vector control staff may be more familiar with, permethrin was detected in about 7% of basins, and then deltamethrin was only detected in 0.5% of basins. So just like a couple, like 2, not very much at all. Generally, we detected pyrethroids. When we detected them, they were found in both phases, the suspended solids and the water. There are some differences in concentration in detection frequency between the media, but not to a particularly significant extent. Permethrin was detected more often in the suspended solids over here. As for why bifenthrin makes up such a huge portion of the detections, previous work has been done, and it's found that bifenthrin, by far, compared to most other common use pyrethroids, has a very, very high half-life, whereas other pyrethroids in sediments may have half-lives ranging from a few days to about 100 days. Bifenthrin in previous work has found to eclipse that at half-life starting at 100 days and going up to way past this graph in 250 or so days, depending on the conditions and the properties of the sediment. And this is due to its multiple chemical isomers making it recalcitrant to degradation, either through abiotic or biotic hydrolysis. Looking at concentrations now, we can see that concentrations seem to decrease. The median of the concentrations seem to decrease by the month, so that corresponds with the reduced detections. Though I'll go over in a later slide, there are a number of outliers in this data set that we are still parsing through, right? In July, in the water, we can see a somewhat downward trend in the maximums of these concentration ranges. And in suspended solids, we don't really see a clear trend, right, in these concentrations. Again, bifenthrin is the primary contributor to these trends because it's pretty much the only pyrethroid that we could get trends from. The other pyrethroids were not really detected enough month to month to be to have monthly plots for. Going over concentrations by region, we can see that there doesn't appear to be a clear, single region that has a vastly greater amount of concentrations compared to the others. at a glance here, it seems like LA has higher residues than some of the other sites, but this is just from the number of basins that we sampled from. And again, we have to look deeper into this to get more concrete information. Again, bifenthrin makes up the majority of the overall concentrations. And you can see a more clear effect over here in LA having greater concentrations. And going back to these outliers, several of these basins have concentrations above what we would have, what we could have expected. Most of these basins, they followed previous literature in that water concentrations went up to about a few hundred parts per trillion, and then suspended solids concentrations went up to about a few parts per million. But in some of these concentrations are above, I don't want to flip back and forth between slides, but some of these concentrations are, 10, 20, 30 times what has been seen in other literature. And these are likely accurate for the samples, but these are not believed to be persistent concentrations, since we did not see anywhere near like same amounts for all 3 months in a single basin. And it's believed that this is due to recent pesticide applications and runoff causing a very high input of the pesticides into the basin. So again, we found that 91% that the vast majority of these catch basins do contain pyrethroid residues, and bifenthrin is the main pyrethroid we want to talk about when we are discussing potential exposure to pyrethroids in these basins. There are other ones of interest. Again, we talk about in vector control for mosquitoes. The significant ones are permethrin, deltamethrin, possibly resmethrin, depending on where you're working, right? But when it comes to environmental persistence, bifenthrin by far is the main one to keep in mind. With this data set, we still need to do a fair amount of work looking at the influence of those outliers on these overall trends, and the influencing factors such as environmental data that we recorded, such as temperature, irrigation, things like that. And then we also do plan on looking at comparing with CDPRs, that's the California Department of Pesticide Regulations database for pesticide use, though we did that with the 2019 samples and did not find particularly useful information, since a lot of that database and a lot of pesticide reporting generally is slanted towards agriculture. There is reporting from vector control districts, of course, and there's supposed to be reporting for private applicators, but for the private and homeowner end, the reporting is not always as consistent. And when it comes to the question of are larvae that breed in these sources, are they getting exposed to pyrethroids? It's most likely that they are being exposed to bifenthrin, at least, though we still, not as much is clearly known about, direct larval exposure and selection and dosage. So we are finishing up that work, and we are also processing results from our follow-up study, which was in 2021 and working in LA County with the help of Susanna and Ernie and other staff at Greater LA Vector Control. We sampled from multiple districts, from multiple structures, in LA County, mostly along the San Gabriel River downstream. These include outfalls, flood control channels, and a few catch basins, though many of the basins this year, for some reason, ran dry compared to 2020. And we sampled from multiple media, including water, sediment, algae, and biofilms, to better understand the partitioning of pyrethroids across these various phases. Certainly with water, there's a lot of information that we can gain from residues, but it's also, important to consider for the larvae their dietary sources, a lot of which can come from algae and various detritus in these water bodies. These are some pictures of the sites to give you an idea. And unfortunately, we laid tiles at every site, but due to the dryness and inconsistency in water levels, there were issues with consistent growth across all of our sites. There was also a high prevalence of duckweed at a lot of these sites too, because these outfalls aggregate a lot of various nutrients and fertilizers, which can stimulate the growth of plants, and as you can see here, and duckweed, which can obscure these tiles. And then also, we are in the process of planning some work looking directly at the possible selection pressure that can come about as a result of the exposure setting that we've hypothesized. We're wanting to look at the effect of possible larval exposure on resistance in adult mosquitoes, such as Culex mosquitoes, right? We need to do some more planning, but basically we want to collect mosquitoes from the environment and compare them to lab strain. And then we want to use environmentally relevant concentrations of pyrethroids. And then for resistance, we want to do bottle bioassays, genotype characterization, and then enzymatic assays. So we have that in the works, hopefully sooner rather than later. And lastly, these are some references. And again, thank you for your time. Sorry if some of this was a little bit unclear, but I hope I, clearly illustrated the what we were working on and its significance. Thank you.
Thank you, Nathan. Very interesting. Questions for Nathan? Feel free to unmute and ask, or you can type questions into the chat. I can start with a quick one. So you mentioned that your basins, some of them dried throughout the season. And I'm pretty sure you also mentioned at the beginning that like the concrete itself can kind of serve as a sink for the pyrethroids. I'm wondering, so like if just say fresh water, like non-pyrethroid contaminated water ended up in those dry basins, would you then expect to find that the pyrethroids would move back into that water?
Yes, that's very likely. And again, these catch basins, the various detritus that they hold at the bottom, such as leaves, sediment, soil, dirt, things like that, even if they run dry, because the basins are generally not quite as hot as the surface, and they don't get exposed to as much sunlight, there is still retention of the pyrethroid residues on that media in the basins, so that when water gets added, it can get pyrethroids can get resuspended in the water.
Okay, a couple of questions from the chat. Is there any consideration of breakdown products?
At the moment, there is not. We know that there is a lot of attention given to, say, 3-phenoxybenzoic acid, which is one of the primary breakdown products of these pyrethroids. But our current work does not look at the concentration of those metabolites in the water bodies.
Right, another question from the chat. Could you clarify what the potential sources of these chemicals are aside from vector control activities?
Okay, yeah. So again, the sprays are, whether it's by a ULV from like a vector control agency or a homeowner or private applicator use, those are again probably the main sources. The vector control public agency applications, those are well recorded, right? Since those are the public agencies in the state database, right? And CDPR's database is considered one of the most robust across the country. But again, homeowner and private pest control company use is not as clearly reported, even though private pest control applicators are supposed to declare use of pesticides. It's likely that it's not one-to-one accurate what they use to what is actually what they declare to what they actually are using. But it's still probably within the urban area. There is some possibility that for some of these urban areas, such as like Visalia or Madera, their proximity to agricultural operations might mean that pesticide drift is occurring, but we aren't really quite seeing like elevated levels in those areas, as opposed to the more definitely urban areas like LA County or Orange County. So that's likely not a significant contributor.
Can I add to that, if I may?
Sure.
Yeah, Nathan is right. In urban environments, we know that a lot of licensed pesticide applicators, they go around, treat around people's homes. And also we have homeowners, and those kind of uses, especially for pyrethroid, they're probably like an order of magnitude higher than for vector control. And also the types of chemicals are different. And for homeowners use or just the structural pest control, you can pretty much get any of those 8 or 9 pyrethroid compounds. And if you go to Home Depot or Lowe's to buy any of the, ant killer and things, they are most likely to contain pyrethroids. For vector control, I think, people are only using 1 or 2 of those compounds, plus pyrethrins. So the type of chemicals are different. And also the use from the other sides, from the professional applicators, from homeowners, which, as Nathan said, is not very well documented. In terms of quantities, probably it's 10 times more, or even many times more.
All right, thank you. A few more questions from the chat. Do you think the chemical that was only detected from 2 places had to do with districts not actively using it, or did all the districts use the chemicals that you tested?
If you're talking about deltamethrin, again, that's I don't, off the top of my head, I'm not quite sure how widely that is used. I know that it is appreciated because it's generally more toxic to mosquitoes than permethrin. And then how many districts? Yeah, so it's likely that, due to its lesser use, that its detection is not quite as high, right? Some of these pyrethroids, they have a higher toxicity to non-target organisms as well. Pyrethroids are, a lot of their environmental concern is because they are fairly toxic to organisms like fish and aquatic invertebrates like little crustaceans, right? So things like permethrin may be preferred. But in terms of all district use, I'm not quite sure. The CDPR database just kind of classes all pest control applications together. So I am not quite sure if I would be able to parse out if a public district is using specific chemicals as opposed to a private applicator.
Makes sense. Celia, should I do 1 or 2 more, or shall we wrap up?
Yeah, sorry if I'm a little slow with these answers.
No, you're fine.
Very fine. We can answer all of the questions before we end this seminar.
Sounds good. Okay. If a pyrethroid is found at a storm drain, what does that tell us about the pyrethroid application in that area? Do we know where the pyrethroid came from? So I would maybe add to that. Do you know like how far away a pyrethroid might be applied that it ends up in a particular storm drain?
Yeah. Unfortunately, our work didn't provide as much information on a lot of the hydrological implications of pyrethroid phaeum transport. A lot of that is necessary to get a better sort of idea into where the pyrethroids after application, where they're flowing into, and how long they're staying in these basins before they get released into the rest of the system. A lot of that depends on things like the slope of like a street, the dimensions of the catch basin, right? Some of these basins, they have drainage holes that are like immediately at their bottom or that go straight to the system. So that explains why water does not pool very high. Some of the other ones have little like ditches or sumps, right? And the hole is a few inches or maybe even feet off of the bottom, right? Which means that water and other detritus can be contained there for multiple periods of time, right? And, I can't, again, these basins, even like down a single street, they can have very widely different dimensions, and that can have significant implications for the amount of loading and the amount of loading and release of the pyrethroids. So as for the exact like area the pyrethroid would come from, it again really depends on the different areas. We found significant differences from we've done sampling in LA County and in San Bernardino County, right? We found differences in the density of like basins across like a single intersection or street, and that would all inform the pyrethroid distribution and transport.
Okay, one last question from Jennifer Hinke. First, she says, great talk. Previous work has shown that some of the pesticides used for ants can be a big component of runoff from houses. She cites Greenberg here. We didn't have a lot of rain in 2019, but would weather be impactful on the samples and what you see in the basins?
Yeah, definitely. And, like you said, the ant pesticide, that's bifenthrin. Bifenthrin, its primary use is considered for fire ants, but it's used for other insects as well, right? And again, it's very environmentally persistent, so that's a main component. In terms of weather, at least in the summer, well, for those of you in California, we had like rain yesterday, so that might affect things, right? But certainly in the dry season from like May to September, usually we, again, in California, don't get much rain at all. So irrigation is really the main thing to look at, like lawn runoff, people watering their lawns, or maybe washing their cars, but that's not that common, right? That would be the main thing to look at in terms of runoff that carries these pyrethroids and runoff that enters these basins. But other impacts of weather are maybe significant. We have the weather station data on things like temperature and humidity, which could impact the samples. So we hope to look at that. Thank you.
Thank you very much, Nathan. Nice job.
All right, thank you to Kirsten and Nathan for presenting today, and Dr. Tara Tiemann for hosting the Q&As. This wraps up our third seminar of the series, so please join us for the next seminar on November 9th from 12 to 1 Pacific time. Have a good rest of your day. Bye.
Thank you. Take care, everybody.